Okay, so I was talking about promoter regions of genes, and what they do is they look at the promoter regions of, let's say, all the genes they can find in bacteria, because they studied bacterial genes first. And then what they did was they aligned, this is just a small section that they're showing on this slide, but they aligned the promoter regions of all the genes they could find and tried to see if there was any way they would line up. And they're trying to find, are there certain nucleotides that occur in that region in front of a gene in every gene they look at. In other words, are there certain nucleotides that are always there no matter what kind of gene it is? And so they, by aligning them and looking for commonalities, they come up with these consensus sequences that they think are important for marking, what a promoter region is for a gene. And so here you have the promoter of a bacterial gene will typically have a consensus sequence centered at what we call -10, which means 10 bases before the beginning of where transcription will start. That on the chromosome you'll have, at this position, you'll have a TA, TAAT. That's a standard sequence there. If any of those gets mutated, you typically won't get transcription of the gene. At -35, you have another consensus sequence, TTGACA. And so there's these little groupings of sequences, consensus sequences, that they have found that are important, and they have to be in the promoter in order for that gene to be able to be expressed. But if those things are there, and then what will happen is the RNA polymerase will come and attach to these things, and that is where it'll start copying. Now, genes and RNA and proteins are all considered to be collinear. The DNA is double-stranded, of course. Transcription copies one strand, and you end up with a strand that looks like this. So, for example, this mRNA starts with a CGU. It was copied from the template strand of DNA that is GCA. But one thing that you'll notice is that the directionality of the mRNA, 5:3, matches the directionality of the non-template strand, 5:3. So, as I was saying before, the mRNA and the non-template strand are both complementary to the template strand, which makes them very similar to each other, with the exception of here in the DNA you'd have a T at this third position, and in the mRNA at this third position you have a U, for example. And so what happens during translation, the mRNA contains the codons, and those indicate what amino acids will be hooked together at the ribosome. So if you get a mutation closer to the beginning of the gene, like in this picture shown kind of on the left, then that mutation will affect maybe one of the first amino acid codons, and it could change what amino acid is added to this end of the protein. If you get a mutation that is more towards the end of the gene, then that will affect one of the codons more here, shown on the right, and it will affect one of the amino acids. You might get a substitution for an amino acid that comes more towards the end of the protein. So collinearity means, depending on where a mutation happens, you'll get an effect on the protein at a equivalent, or a place that is equivalent to where it happens in the DNA. Now, in eukaryotes, we have these things called introns. Introns are segments of what are thought to be junk DNA that are inserted into the middle of a gene, of a coding section of a gene. So in this picture you have exon 1. Exon stands for expressed, or you can think of expressed sequence. Introns are interrupting or intervening sequences. So the introns are the junk. But you have exon 1, 2, 3, 4, 5, 6, 7, 8 exons, but you have these introns that are between them. So during gene expression, after the gene is copied from the DNA into RNA, the initial mRNA, which is sometimes called pre-mRNA, has to be spliced. The introns are spliced out or removed. The exons are spliced to each other, and the final mature mRNA just has the exon copies spliced together. So your mRNA is typically quite a lot shorter once it's been processed or spliced than it is when it's first copied from the chromosome. This is the general structure of an mRNA after it's been processed. You'll have on the 5' end of the mRNA, you'll have some nucleotides, and the codons don't start right away. So you'll have this section called the 5' untranslated region. Then at a certain position, you'll have the start codon, which is always AUG, and that's true in bacteria and in eukaryotes. Then after the AUG, the rest of the letters here will be part of codons until you reach an in-frame stop codon, which is UAG, UAA, or UGA. After that, you still have some more RNA. It's just called the 3' untranslated region. The untranslated regions help with binding to the ribosomes. So this video will be, I'm going to end this section of the lecture here, and you can click on the overview of the processing of the mRNAs.